All right, guys. Today we're going to start the last part of Unit 1, which is on biological psychology. And if we get through everything tonight, that would put your first Unit 1 exam. Your Unit 1 exam would be next week. If we don't get through it tonight and I need 2 weeks to finish Biopsych, that would put your Unit 1 exam would be, let's see, it would be October the 3rd. No, October the 6th. So if we get it done tonight, your exam will be the 29th. If we don't, your exam will be the 6th. So let's play it by ear and see how it goes. So let's start Biopsych right now. So in essence, your nervous system is a complex communication network in which signals are constantly being received, integrated, and transmitted. The nervous system handles information just as the circulatory system handles blood. Psychologists and biologists refer to two divisions of the central nervous system, or actually two divisions of the nervous system, the central nervous system and the peripheral nervous system. So let's take a look. The central nervous system is made up of the brain and the spinal cord, and the peripheral nervous system is made up of all the nerves that lie outside the brain and the spinal cord. It's divided into two parts. The nerves of the somatic nervous system, shown in red, connect to skeletal muscles and to receptors in the skin. The nerves of the autonomic nervous system, shown in green, connect to the heart, blood vessels, other smooth muscles, and the glands. So there are two major types of cells in the nervous system, glia and neurons. Glia come in a variety of forms. Their main function is to support the neurons by, among other things, supplying them with nutrients and removing waste material. In the human brain, there are about 10 glia cells for every neuron. The neurons are cells that receive, integrate, and transmit information. In the human nervous system, the vast majority are interneurons, neurons that communicate with other neurons. There are also sensory neurons, which receive signals from outside the nervous system, and motor neurons, which carry messages from the nervous system to the muscles that move the body. So this is a simplified drawing of a couple of typical neurons. I want to show you the boxed areas because these are the key parts of the neuron. In the front of the neuron here, this branch structure is called a dendritic tree, and each individual branch is called a dendrite. Dendrites are parts of the neuron that are specialized to receive information. Here, this area is called the axon. Axons are specialized structures that transmit information to other neurons or to the muscles or glands. And here, this is the soma or cell body, the part of the neuron that contains the cell's nucleus. Information, as you can see, flows from the neuron through the dendrites and away from the neuron along the axon. So the information is received here, flows through the cell body, through the axon, and then it's sent to another neuron. If we take a closer look at the axon, most human axons are wrapped in a myelin sheath. Myelin is a white fatty substance that serves as an insulator around the axon and speeds the transmission of signals. In people suffering from multiple sclerosis, some myelin sheaths degenerate, slowing or preventing nerve transmission to certain muscles. The axon ends in a cluster of terminal buttons. I'm going to show you a blow-up of what this area looks like. Inside each terminal button are sacs or vesicles containing neurotransmitters. These chemicals serve as messengers that may activate neighboring cells. The junction where the information is passed from a neuron to another cell here, that's called a synapse. So let's take a quick look at this again and do a review. So this would be the axon, this would be the soma, and the terminal buttons would be here and up here. So the neuron fires an electrical impulse called a neural impulse. So in a sense, the neuron at rest is a tiny battery. It's a store of potential energy. As you can see from this diagram, inside and outside the axon are fluids containing electrically charged atoms and molecules called ions. Positively charged sodium and potassium ions and negatively charged chloride ions are the principal molecules involved in the nerve impulse. When the neuron is not conducting an impulse, it's said to be in its resting state. The cell membrane is polarized, negatively charged on the inside and positively charged on the outside. The charge difference across the membrane can be measured with a pair of electrodes connected to an oscilloscope. In a resting neuron, the difference is called the resting potential, and it's about negative 70 millivolts. When the neuron is stimulated, sodium gates in its cell membrane open up briefly, allowing positively charged sodium ions to run into the axon and causing an upward spike on the oscilloscope screen, which you can see here. The change in charge causes potassium gates to open and positively charged potassium ions flow out of the neuron. This outflow of potassium reestablishes a negative charge inside the axon and causes a drop in the oscilloscope screen, which you can see here. The brief reversal in charge difference across the neuron's membrane, which appears as a spike on the oscilloscope screen here, is called an action potential or nerve or neural impulse. And that's all you need to know for today, or actually about the neuron.